Hello friends, good afternoon and welcome to EduSet live lectures. Dear friends, today in developmental biology we will be talking about aging. To discuss this topic we have with us our subject expert Dr. Charu Dogra Rawat. Dr. Rawat is assistant professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you. So the third developmental process, which is post-embryonic, apart from the ones which we have discussed, the metamorphosis and regeneration, it is aging. So it's a post-embryonic process, post-embryonic development, and we call it as development because aging, as everybody knows, is basically the deterioration of the processes, or it's basically going down. So but no growth is involved, but still there is a developmental change that has occurred after the baby has been born and grown up into an adult. So aging constitutes an important post-embryonic development phenomena, and today we discuss about it. So the learning objectives for today's session particularly would be to understand about the maximum lifespan and the life expectancy, to understand the characteristics of aging, to understand the different types of aging, to understand about the model organisms. If we want to study aging, what are the model organisms that we can use, and then understand about the basic concept of aging, the basis why the aging occurs. So aging is defined as the time-related deterioration of the physiological functions necessary for survival and fertility. The maximum lifespan is a characteristic of the species. It is the maximum number of years a member of that species has been known to survive. So every organism or every species has a maximum lifespan. That is, like one individual or one representative of their species might have lived for so long. So that is the maximum lifespan of a particular species, as it is referred to as. For example, Jeanne Calment died in 1998, aged 122 years. Lake trout has the maximum lifespan of 150 years, and so is the tortoise. The dog has the maximum lifespan of 12 to 20 years. The lab mouse survives for 4.5 years, and the Drosophila survives only for 3 months. So the maximum lifespan, which is a characteristic of the species, is the maximum number of years that the species has been known to survive. However, this maximum lifespan can be reduced because of various factors, and that tells us about the life expectancy. The life expectancy is the amount of time a member of species can expect to live. It is defined as the age at which half the population still survives. So it's a characteristic of a population, whereas maximum lifespan is a characteristic of a species. The life expectancy is a characteristic of the population. So depending upon various factors, that the amount of time a species is said to be, or a population is expected to live, is termed as the life expectancy. So life expectancy through the ages is shown here. The early humans did not generally live long enough to develop the heart diseases, cancer, or loss of mental function. So this is how, in different periods the life expectancies has been given. For example, Neanderthals, the life expectancy was around 30 years. For Neolithic, it was 38 years. And for, as we go know that today's individuals have life expectancy of 82 to 85 years, which is there. And in fact, the death used to occur because of various reasons. For example, in Neanderthals, they died of injuries caused by rock falls, hurting accidents and conflicts. Food scarcity led to malnutrition. And these gatherers, hunter-gatherers groups, contracted diseases that spread from animals such as rabies, etc. However, the individuals in the medieval period, the life expectancy grew to 48 to 30 years. And with urbanization, but the famine caused by crop failures and bubonic plagues were the big killers. So the death used to occur because of different reasons, and that is why the diseases which we see today, such as cancer or mental retardation or memory loss, etc., they never used to have those things, because they used to die much before that, much before when such kind of diseases develop. So in today, the deaths are basically due to cancer, heart diseases and strokes are the biggest killers in the developed world. A longer lifespan also comes with unprecedented loss of mental functions and mobility problems. So life expectancy through the ages have been changing, and the reasons for the death is also differing according to the periods. So life expectancy, as you can see, again has the average life expectancy has increased. In fact, in this case, the survival curve, this is for American women. So in 1900s, the 50% of individuals used to be alive till 58 years of age. However, in today's world, it's much higher. It's 70 years or 75 years. It's the 50% of the females, the fertile females, are living. So the life expectancy has increased. The maximum lifespan is one of the individuals, for example, lived for 122 years. So that's the maximum lifespan. But life expectancy when 50% of the population is at that stage. So life expectancy is increasing up to like 80-85 years. The characteristics of aging, if we look at, it's a progressive deteriorative changes which are accumulating, and as it's very commonly observable, the graying of hair, the sagging and wrinkling of skin, the joint stiffness, the osteoporosis, which is basically the loss of bone calcium, the loss of muscle fibers and muscular strength, memory loss, eyesight deterioration, and the slowing of the sexual responsiveness. So these are all the characteristics of aging which is really observable, and it's basically a progressive deteriorative changes that have been occurring. Physiological parameters in aging basically, or the physical inactivity and weightlessness, is accompanied by, for example, reduced maximum oxygen consumption. There is resting and maximum cardiac output. The stroke volume is reduced, the sense of balance is reduced, the body water and sodium declines, the blood cell mass declines, there is a lean body mass which is there, the glucose tolerance test is reduced, the sympathetic activity and neurotransmission is declined, thermoregulation, immune responses. So all these physiological parameters, they become lessened or reduced in their manifestation. The systolic blood pressure and peripheral resistance increases, the vestibular sensitivity increases, the serum total cholesterol increases, urinary nitrogen and creatinine or the urinary calcium also increases. So these are some of the physiological parameters that is increased with aging. Then the endocrine changes or the hormonal changes, they are variable. They may increase or decrease, and they depend upon a lot of different factors, but they become variable. There is an altered EEG electrogram which is there. There is altered sleep, usually associated with insomnia, and there is changes in specific senses which are also observed. So these are some of the physiological parameters that changes while aging, and the person is said to be going into senescence. So this senescence is the constellation of changes in structure and function of an organism that can be observed after sexual maturation. It's a deteriorative change that causes an increase in mortality with the age. So as the individuals grow old, they tend to enter into a senescence stage. And in this senescence stage, practically all the physiological activity slows down, or the structural changes are there which occurs. These kind of changes, or the changes that was occurring during the senescence, prompted Shakespeare in As You Like It to actually say that those who did survive to senescence left the world sans teeth, sans eyes, sans taste and sans everything. So basically a person who is able to go up to the senescence leaves the world without everything that he was born with. So that's how he puts up, and that's how he portrays the essence of the senescence that occurs during the developmental process of an individual. Looking at the different terms which are used for types of aging, there is chronological and physiological aging. So chronological, as the name suggests, is determined by the passage of time since birth. However, the physiological aging, or also called as the biological age, is determined by physiology of an individual and is dependent on many external factors. So chronological age is the time dependent age. With the passage of time, the individual is going to grow old, and all the activities are going to slow down, but they are all related to time. With the passage of time, these things occur. So that is defined as the chronological age, and it is calculated in terms of years. For example, a person is 30 years born, or 40 years, or 50 years of age, which is basically the time that has elapsed since his birth. Physiological age is, however, the status of the body of an organism. So the status of the biological age depends upon the physiology, the functioning or the working of the body of the individual, and it is dependent upon many external factors. The two terms which are again used in the context of aging is the intrinsic and the extrinsic aging. So intrinsic aging is basically the chronological aging, time defined aging, which is basically because of the intrinsic factors, that there are some changes which are time bound that will be occurring, and therefore it is termed as the intrinsic aging. The signs usually include the drying of the skin, the wrinkles, the large pores, redness, and decreased healing capability. However, extrinsic aging is basically that is influenced by the external factors. The signs include, in addition to the natural signs of aging, the increased roughness, or the spots are found everywhere. That's because of taking some kind of taking not a very healthy food, or going out into the sun, and that causes excessive wrinkling or excessive spotting, etc., or the sagging of the skin. Naturally there is some kind of sagging, but depending upon the external environment and conditions, there can be much more severe sagging of the skin that can occur. So this is termed as the extrinsic aging, and these are the signs of the extrinsic aging. Two another terms are used which are called as the primary and the secondary aging. So primary aging is basically the progressive deterioration in physical structure and biological function that occurs with advancing age alone. So chronological aging or intrinsic aging can also be called as the primary aging, which is depending only upon that the time has elapsed, and after so much amount of time these changes are going to occur in the body, and therefore the process of aging is occurring, and it is independent of any other external or internal factors. It's basically time bound, and after a particular time has elapsed, the body needs to it's wear out, or it's going to age. So that's called as the primary aging. Secondary aging is the accelerated deterioration in organ structure and function that is mediated by diseases such as diabetes and hypertension, or by harmful environmental and lifestyle factors such as tobacco smoking or excessive sun exposure. So secondary aging is basically extrinsic aging, that is dependent upon the external factors, the environment which we live in, the environment which we subject our body to live in will deteriorate. The progressive deterioration in organ and structure would occur, and that is termed as the secondary aging. For example, the lifestyle diseases, the diabetes and the hypertension, or the excessive smoking or tobacco use or alcohol use, they are all going to affect the body's physiology, and therefore it's going to age more quickly, or accelerated deterioration will occur, and such a kind of aging is termed as the secondary aging. Then there are certain key facts, the aging and the health, related to the aging and the health, and the World Health Organization has released a fact sheet in September 2015, because as we have seen, life expectancy is growing. Life expectancy is growing since the historic ages. Now a person can live up to 80 to 85, on an average can live up to 80 to 85 years. But the thing is that this aging phenomena, it's inevitable, it is going to occur. There is a chronological age, but then individuals are making it more of a secondary aging. They are using such factors that tends to enhance the secondary aging. So the health is very important. Health during the aging time is very important. So World Health Organization came up with these fact sheets. They find out the key facts that the aging and health are related. So during the process of aging, how the health can be maintained so that to have a good senescent period. In any case, every individual needs to age and die, But then during this, because the life expectancy has increased, therefore the person stays in this aging period right from 40 to probably 80 years. So how can this progressive deterioration can be made easier, or made it's a healthy aspect be given to it. So some of the key facts I'll just talk about first, that between 2015 and 2050 the proportion of the world's population over 60 years will nearly double from 12% to 22%. So as I said, because the life expectancy is increasing, and moreover the people are going towards the age, so 12% of the individuals which are over 60 years now, that proportion will double, and 22% of the individuals of the world's population will be over 60. So by 2020, the number of people aged 60 years and older will outnumber the children younger than 5 years. In 2050, 80% of old people will be living in low and middle income countries. So when initially the aging process started with the developed countries, but now by 2050, 80% of the older people are expected to be living in the low and middle income countries. The pace of population of aging is much faster than in the past. So in the past that we have seen that the people might not be aging because they never used to live that long. Moreover, because of all these external factors, the secondary aging or physiological aging or extrinsic aging has accelerated. So because of major uses or the lifestyle disorders and everything, this aging process has been fastened. And as it says, the pace of population aging is much faster than what it was in the past. And therefore, all countries face major challenges to ensure that their health and social systems are ready to make the most of this demographic shift. So because now the entire the population is becoming more and more aged, there are more individuals of 60 years and above the age, and this aging process is also accelerated. So as a countries, every country as a society has this liability to ensure that their health and social systems are ready to make this kind of, or ready to handle such kind of aging phenomena that is occurring. So for those individuals and for those things, in accordance with the recent World Health Resolution, a comprehensive global strategy and action plan on aging and health is being developed by WHO in consultation with member states and other partners. The strategy and action plan draws on the evidence of the World Report on Aging and Health and builds on existing activities to address 5 priorities areas of action. And these 5 priority areas are the commitment to healthy aging. So as I said, aging is an inevitable phenomena. The chronological aging is an inevitable phenomena. The developmental process which stops and the individual is born and then it grows into an adult, then slowly and steadily this will go down and the aging will occur. So this is an inevitable thing. But because the life expectancy has increased, the person staying in the senescent stage is much more, the time span spent in this senescent age is much more. Therefore, it's important to ensure the health during this aging process. So the global strategy and action plan of WHO, the first priority area is the commitment to the healthy aging. Then aligning health system with the needs of the older population. So health systems is very important, So there has to be many more health systems that address to the needs of the older population, as we have seen that older populations are gradually increasing. Then developing systems for providing long-term care. Now, since they are spending much more time in this, much more time span in this aging time period, then we need to, the countries need to develop the system for providing long-term care. Then creating age-friendly environments, because now there will be more of the older people, so there has to be, more care and more friendly environments for them. And then improving the measurement, monitoring and understanding constitutes the priority area, that to understand about the needs of the older populations, to measure and monitor the process of aging also, that how it is being going on and how it sustains and how the individuals are going to stay in the aged time span which is there. So these are the 5 priority areas which they are talking about, and so basically these are the social issues or social concerns about the aging that I just wanted to point out. Now, how this aging can be studied? This aging has a complex phenotype. The studies on the humans are very difficult because it's slow, the longer lifespan of the individuals. It's expensive, then genetic variability, environmental variability, then there are certain ethical issues also. So just studying aging in the humans, it's a very difficult task. So what you need is the need of the model organisms. The model organisms are small, they are comparatively inexpensive, they are experimentally tractable. The factors that may affect aging can be experimentally manipulated. They have a shorter lifespan, as we saw that mice has around 4 to 4.5 years. Even in fact, Drosophila only has a few weeks of lifespan, 3 months of lifespan. Then controlled environment, we can put the model organisms in. We cannot raise the humans in the controlled environment. And minimize genetic variation by using inbred animals. So animals we can inbreed, amongst themselves, amongst their own relatives, and therefore create pure strains. And it's much more, when the background heterozygosity is minimal, then it's very fruitful to study any kind of change that is occurring because of one phenomena. In the humans, the genetic variability is far more, and therefore it's very difficult to understand the phenotype of the aging. But since the animals we can raise the inbred strains, it becomes much more handy to carry out the aging in these kind of model organisms. So some of the model organisms are, for example, yeast, the Saccharomyces cerevisiae, the roundworm, the C. elegans, the fruit fly, Drosophila melanogaster, and the mouse Mus musculus. So these are the model organisms that are used for the process of aging, and some of the underrated models of aging are the rapidiles, naked mole rats, and whales. They have been also used in some of the studies for understanding aging, but these are usually the underrated models, and the yeast, roundworm, fruit fly or mouse are the most important model organisms that are used. Now look at the shared phenotypes between these model organisms. Why and why can we use these model organisms, and can we compare it with what happens in the humans? So the shared phenotypes is that the aging process increase in the mortality rate over time. So as the aging occurs, there is increase in mortality, and this happens in all of the model organisms. Then there is a stress resistance decline. So the organismal and the cellular level, which is observed with several different stressors such as heat stress, oxidative stress, hydrogen peroxide stress, high oxygen, heavy metal stress or osmotic stress. So under all these stress conditions, during the process of aging, the stress resistance declines both at the organismal as well as the cellular level in all the model organisms. Then physiological function declines with increasing age. So in all the model organisms, generally conserved to the extent that the physiology is conserved. For example, in worm, fly, mouse, rat, they have muscle cells, and a decline in muscle function is observed at these animals' age, modeling the decline in muscle and sarcopenia in humans. So the muscle cells exist in all the animals, and the muscle decline or the muscle damage, deterioration occurs in all these animals, and it can be modeled to compare it with the human's muscle decline and sarcopenia, the age-related muscle deterioration in case of humans. Then the diseases of aging, if we look at, the mouse and rats are the closest models. The heart disease, cancer, arthritis, kidney disease, neurodegenerative diseases, all these diseases of aging are can be found in the mouse and rats, and therefore compared to the humans. Some aspects, some diseased aspects can be modeled in fly and worm also. For example, models for Alzheimer's disease and some other types of neurodegeneration or aspects of heart diseases are seen in the fly. So that makes it easier for us to use model organisms and understand the aging phenomena in case of humans. Then cellular changes in the aging are also similar in these organisms. The nuclear changes, that includes the nucleus enlarges, The nucleolus changes morphology and undergoes fragmentation. There is reduced efficiency of DNA repair. The total gene transcription is lower. There is an altered in gene transcription. The protein turnover declines. There is advanced glycation end products which are formed. So, all these cellular changes are also accompanied during the process of aging in all these model organisms. So, these model organisms share these phenotypes with humans, and therefore, it is easier, and because of the reasons, I already told that they have shorter lifespan, they can be experimentally manipulated, etc. The model organisms are used to understand the mechanism of aging. Now, looking at why the aging occurs, there is an evolutionary theory which defines that why does the aging occurs. The Huntington's chorea disease, for example, it's a genetic neurodegenerative disease caused by a highly penetrated dominant mutation. In 1948, Heltane asked that why has natural selection not acted to remove the Huntington's mutation from population. So, we already know that the evolutionary, the evolution always select the things which are advantageous. They always select upon the things which are advantageous for a population, and therefore, it sustains. But Huntington's chorea disease, it's a highly penetrant dominant mutation. So, in 1941, Haldane asked that, why doesn't it was not, selected upon, and why the natural selection did not act against it and did not select or removed Huntington's mutation from the population. The reason could be that the average age of onset of Huntington's disease is 35.5 years. And as we saw in the life expectancy, for much of the evolutionary history of mankind, most people did not even survive up till that age. It was 40 years life expectancy up to medieval period. So, the individuals used to die much before that, and therefore, the disease never came up. And because the selective pressure to remove this disease was therefore weak, there was a giant conceptual leap. Perhaps that is what aging is. It is the result of late acting deleterious mutations, the associated diseases with the mutations and everything, they are basically late acting. So, even in a population free of aging, the death will nonetheless occur from extrinsic hazards such as diseases, predators and accidents. So, with time the age will or the death will occur, and it is said that recurrent deleterious germline mutations, few bearers survive to express the later acting mutations. As you can see in the later half of the graph, very few individuals are living. So, the force of natural selection against these deleterious mutations is very weak, and therefore, these mutations can reach a higher frequency under the mutation selection balance. So, there is an early acting mutation, most bearers are still alive, and therefore, there is a strong force of natural selection and it will be removed. But the age related diseases, they generally are late acting mutations, few bearers are alive, and therefore, there is a weak natural force of selection that occurs. And another way of looking at it is the pleiotropy or the trade-off theory for the evolution of aging is also stated, that suppose there are mutations that are beneficial in the youth, but at the price of a higher rate of aging. So, more individuals will survive to express the early benefit than will survive to suffer the higher rate of aging. Mutation like this can never therefore be incorporated by natural selection. So, aging evolves as a side effect of natural selection in favor of mutations that cause a benefit during the youth. So, either the early acting mutations or any mutations that are affecting the advantages for the youth is basically selected upon, and the late acting mutations, they are weaker forces of natural selection, and basically maybe they are not that much, and therefore, the process of aging gets manifested in the populations. So, after understanding the concept of aging, that what aging is, what are the structural and physiological changes associated with aging, what are the different types of aging, and then how the society needs to respond to this aging and health, we will look at the basis or the mechanism behind the aging. What are the theories that explain this phenomena. So, the learning objectives for this session would be to understand about the various theories of aging, the programmed and the error theories, to understand about the genetic aging, and the basic concept of the basis of aging which we will talk about will be these. So, looking at the how of aging, two main aging theory categories are there, the programmed theories and the error theories. So, programmed theories, aging has a biological timetable or internal biological clock. So, as we talked about the chronological age, there is a biological clock that after so many years have elapsed after the birth, the deterioration needs to start. So, there are certain theories which explain this kind of aging, and then there are certain theories which are called as which are summed up under error theories, that the aging is a result of internal or external assaults that damage the cells or organs so that they can no longer function properly. So, they emphasize on the damage that occurs because of intrinsic or extrinsic factors. So, these are termed as the error theories, and usually many theories are a combination of both the programmed and the error theories. So, programmed theories include the programmed Senescence theory, the endocrine theory and the immunology theory, and the error theories include the wear and tear theory, the rate of living theory, the cross-linking theory, free radical theory and the error catastrophe theory and the somatic mutation theory. So, looking at the programmed theories, we will first take up the programmed Senescence theory. The programmed Senescence theory is the result of sequential switching off or on of specific genes. So, it is basically after the birth, when the individual has grown into an adult, then there is a sequential switching off or on of specific genes based upon the time, and that is termed as the programmed, the programmed apoptosis, the programmed cell death which is called as. So, after certain cell divisions, the cell needs to die, and that basically constitute this. For example, this is termed as the Hayflick limit, that you understand that cell division, it follows a cell cycle. There is a G1, G0, G1 phase, there is a S phase, there is a G2 phase, and there is a cell division phase, there is a replication phase in between, and the cell keeps on undergoing this cycle and producing more of itself. Then one of the cells may enter into a G0 phase, which is the resting stage, but after a certain number of cell divisions, which is termed as the Hayflick limit, it enters into the G0 or the quiescent stage, and it is irreversible. It cannot come back to the cell cycle stage. So, it remains in this G0 or quiescent stage, and that is basically when the senescence has occurred. So, Hayflick limit is the maximum amount of number of times a cell can divide in a proper normal manner. After that, it will go into the G0 stage, an irreversible G0 stage basically. The fibroblasts are removed from umbilical cord and cultured. The fibroblasts divide and repeated until approximately 50 divisions, and it will not divide past this point. So, this Hayflick limit is telling us about the programmed cell death, that after a certain amount of divisions, the cell will get into a stage where it will not, where everything will stop, the cell division will stop. So, it will enter into a quiescent stage or a G0 stage, and the cell will cease to divide. This is a programmed senescence. This is a programmed senescence theory, or this is called as the programmed, and then when it enters the senescence stage, slowly it dies, and it is called as programmed cell death or apoptosis, which is there. Another theory which explains this kind of programmed thing is the telomeric theory. This is an extension of the Hayflick limit. Telomeres, as you know, they are specialized DNA sequences at the end of the chromosomes, and they shorten with each cell division. When the telomeres become too short, the cell enters the senescence stage. In the normal process of DNA replication, the end of the chromosome. So, from molecular biology, you already have this idea that in the normal process of DNA replication, the end of the chromosome is not copied completely, and there always occurs a gap. When a human cell divides, its DNA splits into two strands, one with a gap at its end. that end gets shorter after each division until the cells can no longer divide. So this telomere will keep on making the DNA shorter and shorter. However, there is an enzyme which is called as the telomerase enzyme that lengthens the strand opposite the gap, and another enzyme then fills in the gap, keeping the strand at youthful length and allowing the cell to keep dividing. So if this gap is not filled, then the telomere will keep on shortening after each cell division, and the cell is said to will die eventually. But telomerase enzymes maintain this. So telomerase theory explains that with age, the telomerase enzyme reduces in number, and therefore the telomere becomes shortened and enters into a senescence stage. The enzyme telomerase fills the gap by attaching bases to the end of the chromosome. As long as the cells have enough telomerase to do the job, they keep the telomeres long enough to prevent any important information from being lost as they go through each replication. However, with time the telomerase levels decreasing, and this decreasing telomerase levels, the telomeres become shorter and shorter and eventually lead to the process of senescence and aging and eventually the death. So this is the telomerase theory. The shortened telomeres are found in some of the diseases such as atherosclerosis, heart diseases, hepatitis and cirrhosis. They have been delivered, and in fact telomeric theory and cancer can also be associated. 90% of the cancer cells have been found to possess this telomerase. Telomerase prevents the telomere from shortening, and this allows the cancer cells to reproduce, resulting in telomeric growth. So because of the cancerous growth or unprecedented cell division, it is found that presence of telomerase ensures that the telomere do not get shortened and therefore do not enter the senescence stage, or the aging does not happen. Therefore, the research areas focus on measuring the telomerase may help detect cancer. Stopping telomerase may fight cancer by causing death of the cancer cells, and telomerase may be used to help with wound healing or the immune response. So telomerase enzymes, the quantification of the enzyme in the cell can tell the state of the cell, whether it is young or whether it is going to die or it is an old cell. So this is the telomeric theory of aging. Other programmed senescence theory is the endocrine theory. So endocrine theory is basically states that the biological clock acts through hormones to control the pace of aging. Hormones affects growth, metabolism, temperature, inflammation and stress. So there are endocrinal changes or hormonal changes that are associated with aging, and this biological clock, it acts through these hormones, and because hormones are usually the signaling molecules, it signals that the aging has started. For example, the menopause, the decreased levels of estrogen and progesterone are there, and there are hot flashes and insomnia occurs in the menopause stage. So endocrine theory is based upon such changes that are influenced by the alteration in the hormonal level in the body, and which is then associated with the process of aging which is there. Immunological theory states that a programmed decline in the immune system leads to an increased vulnerability to disease, aging and death. For example, there is a decreased T helper cells in adults, increased diseases in older adults and increased autoimmune diseases in adults. So immunological theory focuses on the weakening of the immunological system which is programmed with age. So as the aging or as the senescence continues, the immunological processes, T helper cells etc., they are reduced in number, and therefore the body is affected by many diseases which are the characteristic of aging period. So this is what is explained by the immunological theory. Then there are a lot of error theories. As we said, instead of the biological clock, there are many intrinsic and extrinsic factors that play a role in this aging process, and they comes under this error theories. The first is the wear and tear theory. The wear and tear theory states that the years of damage to cells, tissues and organs eventually wears them out, killing both them and the body. So when the cell is dividing and when the cell is forming into tissues, then years and years of their usage wears them off, and eventually they start dying. So after a certain period of time, they start losing its functionality, they start, the damage starts to occur. So there is a wear and tear that is occurring to the cells and the tissues, which is damaging them. For example, the wearing out of the skeletal system, such as in osteoarthritis. So with age, as they say, that that the bone minerals are lost, and these minerals are lost, or the knee area, basically because of heavy usage or overall usage and the decrease in the calcium, etc., this osteoarthritis can occur. So wear and tear can be viewed as a result of aging basically, and not the cause of it. So looking into this theory, it's better to explain this wear and tear thing as a cause of aging or a result of aging, rather than stating that because of wear and tear the aging is occurring. But that is one of the theories that is there. Then the second theory is the rate of the living theory. Rate of living means the metabolic rate. The greater an organism's basal metabolic rate, the shorter is the lifespan. Free radicals or other metabolic byproducts play a role in senescence. So this is again one of the theories which state if an individual has a higher metabolic rate, then they tend to age easily, or they tend to have a shorter lifespan. The free radicals or other metabolic byproducts play a role in senescence. For example, animals with the most rapid metabolism tend to have the shortest lifespan. That is, birds have a shorter lifespan than humans. Studies examining the relationship between metabolic rates and longevity, however, have produced inconsistent results, limiting the usefulness of this theory. Since we are discussing all the theories related to aging, I have included this rate of living theory. But there is not substantial evidence which actually states that the ones with the higher metabolic rate or higher basal metabolic rate tends to be longer, because athletes usually have a higher metabolic rate, or some people with particular occupations will have a higher metabolic rate, but it does not correlate with the longevity of those individuals or such individuals. Therefore, there is no evidence for it, but that was supposed to be one of the explanations for aging, that probably the one with the higher basal metabolic rate have shorter lifespan. Another theory is the cross-linking theory, and this states that the accumulation of cross-linked proteins damage cells and tissues, slowing down bodily processes. So again, there is an accumulation of certain substances which might be toxic to the cells and the tissues. The cross-linking protein products, they might damage the cells and tissues and therefore lead to the process of senescence. For example, non-enzymatic glycosylation reactions occur when glucose molecules attach to proteins, causing a chain of chemical reactions resulting in a structural change to the proteins. So there is loss of flexibility of connective tissue, and microvascular changes in the arteries are also there. So such cross-linking proteins will slow down the bodily processes, and such chemical reactions, will cause the structural change to the proteins, causing the damage to certain tissues and the cells. And this is the cross-linking theory which we talk about. Then a very important theory that explains that attempts to explain the process of aging is called as the free radical theory. During aging, the damage produced by free radicals cause cells and organs to stop functioning. So what are free radicals? A free radical is any atom, molecule or ion with an unpaired valence electron. So you can see there is an electronic configuration of an atom which is shown. There is a nucleus and there are orbitals, and in the orbitals there are electrons. Then electrons tend to live in pairs, but there is one electron in which there is a missing electron is there, and therefore it creates a free radical, and this free radical is highly reactive because electrons love to remain in pairs, and therefore they are highly reactive species. One type of highly reactive free radical is the oxygen free radical, which may be produced during metabolism or as a result of environmental pollution. Oxygen free radicals are formed in our cells naturally during the oxidation of food to water and carbon dioxide. And where does this happen? In the mitochondria. So mitochondria, there is a lot of free radicals that are produced because of the oxidation of food to water, etc. These free radical grabs electron from any molecule in its vicinity, and it does so because the electrons like to exist in pairs, and when it grabs an electron from another molecule, it damages the other molecule. So, how this free radical tends to create damage in cells and tissue is by the fact that because they do not have paired electrons, they tend to snatch one extra electron from the tissues that are in its vicinity, or the cells in it, or basically the atoms in its vicinity. And in a process of doing so, they convert them into a free radical, or they damage their sanctity and cause the damage which is attributed to the process of aging. Some of the molecules that may be damaged by free radicals are fats, proteins and DNA, both in the nucleus and the mitochondria. You know, oxygen for free radicals are produced in the mitochondria, but other free radicals can be produced in the nucleus, etc. also, or they can be diffused into the nucleus. If membrane fats are attacked, then you get the breakdown of the cell membrane. If it is a red blood cell membrane, hemolysis occurs. If proteins are attacked, you get the breakdown of the proteins, which may result in the loss of biological function and accumulation of catastrophic compounds. And if DNA is attacked, you will get a mutation that may cause again the cancer, or the aging phenomena is again manifested. So, these free radicals are very dangerous. As you can see, the free radical green attacks the membrane, it can release another type of free radical which is blue. This is the damaged membrane it has already caused. Then this blue one will attack the DNA, releasing another free radical. So, you know, it's kind of a chain reaction. One free radical will produce another free radical, and in the process it will keep on damaging the fats, the proteins, the membrane, the DNA, etc. It will keep on damaging those things. If they are so dangerous, then why does the cell does not take care of it? So, the free radicals do not go unchecked. The body has a multi-layered defensive system that reacts and detoxify the damaging radicals. The defenses include such as the natural antioxidants in the body, such as bilirubin, enzymes such as superoxide dismutase, catalase and glutathione peroxidase, dietary antioxidants such as beta-carotene and the vitamins C and E. So, under normal conditions, your natural defense mechanism prevent most of the oxidative damage that is occurring. However, the free radical theory of aging proposes that little by little small amounts of damage accumulate and contribute to the deterioration of tissues and organs. So, although these free radicals are really dangerous entities, but they do not cause so much of damage because they are always kept under check. There are antioxidants which absorb these free radicals and they give their electrons and make them paired and does not let them loose to cause the damage. However, there is still little damage that can occur, and accumulation of that little damage with time can lead to the process of aging or the deterioration of tissues and organs which are there. So, this diagram particularly shows that how an antioxidant molecule will take care of the free radical, and then the membrane will be repaired and the DNA is damaged, but the DNA remains damaged, impairing the cell's function. In addition, the antioxidant molecule now has an unpaired electron and thus becomes a new radical. So, I always told you that this free radical generation is a chain process. For example, then when vitamin E scavenges free radicals, it becomes a free radical by themselves and may be more carcinogenic than the original free radical. This is the reason why taking high doses of vitamin E supplements appears to increase cancer risk in a person, not decrease the cancer risk. So, it is very important to understand that free radical generation is a chain process. The antioxidant will give its electrons to neutralize the free radical, but in turn themselves become a free radical, but they may not cause damage because they may not be large in numbers, but then therefore it has to be taken, the dose has to be kept under control, and little damage that keeps on accumulating might lead to the occurrence of the deterioration of the tissues and the cells in the body which is there. Another theory that explains aging is the catastrophe theory, and the catastrophe theory states that any damage to the enzyme systems that synthesize proteins in the body results in faulty protein synthesis. The faulty proteins continue to accumulate in the cell until they reach a level that damages the cell's tissues and organs. When enough damage accumulates, this may result in cell malfunctioning leading to the death. So, catastrophe theory basically states that any error which is there will lead to the formation of altered protein products, and these altered protein products, if not removed, will keep on accumulating inside the cell, and keep on accumulating inside the cell. They will create a kind of a catastrophe and they will damage the entire tissues and organs, leading to the process of aging which is there. Then there is a somatic mutation theory. The genetic mutations occur and accumulate with age in the somatic cell, causing the cell to deteriorate and malfunction. The accumulation of mutations results in the damage to the DNA. The theory states that aging is an imbalance between DNA's ability to repair itself and accumulating DNA damage. When the damage exceeds the repair, the cell malfunctions, and this can lead to senescence. So, that there are the errors which are made while replication or transcription leading to the mutations. They are generally repaired and they are checked upon. But sometimes they can keep on going, and when the damage exceeds the repair phenomena, then the cell can malfunction, and this can lead to the process of senescence which is there. So, accumulation of mutations can result in. So, the natural the spontaneous errors that can occur in the DNA, etc., they can accumulate and lead to the process of aging which is there. Going deeper into the genetic aging, the genes have been shown to affect aging. So, we were talking about different phenomena or different error theories or programmed senescence theory in which the systems were affected, the physiology was affected, the cells and tissues were damaged, leading to the aging. Now, we look at that there are certain genes that have been attributed to the aging. So, functioning or dysfunctioning of certain genes are associated with the phenomena of aging. In a very good example of this syndrome called as Hutchinson-Gilford progeria syndrome, the children ages rapidly and to die usually of heart failure as early as 12 years. So, this is a very unusual syndrome in which the children tends to age rapidly. So, although they are of the person shown in the figure might be just of like 10 years of age, but he looks like a 60-year adult. So, they tend to age rapidly and die usually of heart failure, etc. It is caused by a dominant mutant gene, and its symptoms include thinning of skin with age spots, resorbed bone mass, hair loss, and arteriosclerosis. So, these are the phenomena which are associated, or these are the characteristics of aging. So, these characteristics of aging are seen in a child, and that is basically the Hutchinson-Gilford progeria syndrome. Now, a gene was found to be associated with this syndrome, and this gene is called as the Klotho gene. The Klotho gene, as you can see, the wild type gene is basically active gene, and it remains as the age of the individual to be shown as whatever the age is. The Klotho mutant, however, suffers from premature aging and age-related disorders due to a deficiency in the normal Klotho protein. So, this Klotho protein is very important for the normal functioning of the cell or normal phenomena, various physiological phenomena of the cell. However, the Klotho mutant, the aging process is much more fastened. For example, in this you can see that there is a normal mouse and there is a Klotho deficient mouse which is there. Then in one of the mouse there is an overproductive Klotho gene is also there. The mouse deficient in the Klotho gene shows signs of aging compared to the normal mouse, and the 3-year-old mouse on the right has lived beyond the normal lifespan. So, if the Klotho gene is overexpressed, then the mouse has past the lifespan of the mouse, therefore it has it is living for more than 3 years. So, aging has been delayed if there is an overproduction of the Klotho gene. However, the Klotho deficient mouse is showing the symptoms of aging much before the aging age has been acquired. So, this is one of the gene that has been attributed to cause the aging. In C. elegans, the model organism which we talked about, there are 2 genetic pathways that affect aging. So you can see that there is a wild type individual. In the wild type individual, there is an adult development, and the adult is usually a short-lived adult which live only for few weeks. However, if there is a lot of overcrowding, or there is a food starvation, then this starved wild type can enter into a state which is called as the Doyer state. The development of C. elegans contain 4 instar larvae, after which this shorter lifespan adult is born. But during any of the larval stages, if there is overcrowding or there is starvation, then they may enter into a stage which is called as the Doyer stage. Then in the Doyer stage, the Doyer larva will remain up to 6 months, and if this larva is then if this larva gets the right conditions again, then it is going to develop into a normal short-lived adult, which is basically there. However, in the mutant, if there are certain mutants in which 2 of the genes such as DAV2 and AGE1 which are deficient, they do enter the Doyer stage, and the adult that develops then lives a longer lifespan than the wild type one. So these 2 genes can be attributed to the aging phenomena, and therefore this pathway is shown that there is an insulin like protein receptor which is there, and there is a DAV2 receptor. So this protein, when binds to it, creates a signaling pathway, and this signaling pathway leads to the short-lived adult development. In the wild type starved stage, there is no stimulation which is there, and therefore adult development is halted, and the larva enters into the Doyer stage. These Doyer stage will live as a longer lived Doyer larva, and ultimately will give rise to the same short-lived adult. However, in the mutants which are DAV2 or AGE1 deficient mutants, the adult development is the adult which comes out of this Doyer stage lives has a very higher lifespan than even the wild type mutant which is there. So these genes play a role in in altering this Doyer stage and increasing the lifespan. The second pathway basically involves the gonads. The germ cells appear to inhibit longevity, while the somatic cells of the gonads act to prolong the life of the nematode. So this is again one of the indication in the case of C. elegans, that germ cells appear to inhibit the longevity, while the somatic cells of the gonads act to prolong the life of the nematode which is there. So depending upon knowing about all these theories tells us about that how the aging phenomena is occurring. Although not only a single theory explains that this is the way the aging occurs, but it's usually a mixture of many theories that constitutes the basis of the aging phenomena. And despite the fact that there are genes which are responsible for causing this aging to occur, but this is the most important post-embryonic developmental process that occurs after the birth of an individual. Thank you.
On that note, I would like to thank ma'am for this very enriching discussion, and thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.